Hi, today in this video we are going to see how to draw a bacterial cell. So we begin that by drawing the outer boundary for the cell. So we draw that in a rectangular shape but with rounded corners. And you don't really have to be very perfect with this, just go with the flow. So this is the um, cell wall of the bacteria. Right, and within the cell wall is found the cell membrane. So we draw that by just uh, following the cell wall, but inside. So we are basically drawing a smaller rectangle with rounded corners. Now, as you can see, I'm leaving a gap between the cell wall and the cell membrane. This means that they are not stuck to each other, they're separate entities, and so just when you're drawing, leave a gap. So this we label as the cell membrane. Now these two boundaries enclose what is called as the cell. So the interior portion of the cell is called as the cytoplasm or the cytosol. So this is an aqueous fluid which contains all the cell's components. Now the next part of the cell is the DNA. So as you all know, DNA is the hereditary material, and in the bacteria the DNA is found directly in the cytosol. So the region containing this DNA is called as the nucleoid. Now as you can see, I have just drawn a squiggly scribbled line for the DNA. So this is how easy it is to represent the DNA. And the region that the DNA is found in the cytosol, that surrounding area, is called as the nucleoid. Nucleoid. That is, it is similar to the nucleus, but it is not exactly a nucleus because it does not have a membrane. So this is the major DNA, or it is also called as the chromosome. Chromosome. But in many bacteria, there are some other smaller DNA particles found, and these are called extra chromosomal DNA or plasmids, and we represent them by tiny circles. They are actually circular DNA, so this is how we draw them. Now these can be numerous or none or quite a few, so we just show a random representation of these plasmids. So let us label these as plasmids. So these basically confer some special characteristics to the cell, such as antibiotic resistance or the presence of pili, which we will see in just a few minutes. Now another part of the cell is the ribosome. So this ribosome, which we represent using small dots, is the machinery which helps in the production of the proteins. So DNA codes for the proteins, and the ribosome is the machinery which converts all the complex text that is in the form of DNA into proteins. So these are basically numerous in number, and they are distributed throughout the cell. So when you're drawing, be sure to just evenly distribute these ribosomes throughout the cell. So let us label these as the ribosome. Right, so these are the contents that are found inside the bacterial cell. Now as I said, I mentioned a word called the pili. So a pili is a thin hair-like appendage that is found extending from the plasma membrane through the cell wall into the exterior. So this is a hair-like structure that helps the bacteria in exchanging DNA. So these are just hair-like structures, and they are called pili. Right? These are found throughout the cell, but I'll just move on to another structure, which is called as the flagella. So this also extends from the plasma membrane, and this is used for locomotion. So this is the flagella. Flagellum singular, flagella plural. So the flagellum is found generally on one side of the bacterial cell or two sides. It's not so numerous as the pili, which completely surround the cell. The flagellum helps in the locomotion. So bacterial movement is helped through this flagellum. And again, this is not found on all bacteria. It is found only on certain bacteria, and the same is the case with the pili. So this is a general, this is how you go about drawing a bacterial cell. Now I will just show you a completed bacterial cell. So this is how your complete cell should look like. It's fully covered with the pili. There is a flagellum on one end, the nucleus, I mean the nucleoid region containing the DNA, plasmids, the ribosomes, cell wall, and the cell membrane. So you have now learned how to draw a bacterial cell.